This is our second and final chapter on animal reproduction. In the last lecture, we looked at reproduction in general. This time we're going to look at focus in on human anatomy as well as gametogenesis. We'll start with the male reproductive anatomy. The male's external reproductive organs are the scrotum and penis. The internal organs are the gonads. The gonads are the organs that create the gametes. They also generate important hormones, such as testosterone. There are also accessory glands that secrete products needed for sperm movement, and there are ducts that carry the sperm and glandular secretions. The male gonads are called the testes. These produce sperm in highly coiled tubes called seminiferous tubules. Production of normal sperm can't occur at body temperatures in most mammals. Therefore, the testes in many mammals are held outside the abdominal cavity in the scrotum, where the temperature is lower than the core body temperature by about 2 degrees Celsius. Furthermore, male gonads in humans descend just into the scrotum just before birth. From the seminiferous tubules of the testis, sperm pass into the coiled duct of the epididymis. In humans, the total time for production of a sperm cell is 7 weeks. 3 weeks of this time is spent traveling the length of the epididymis, which is about 6 meters. During this time, the sperm become mature and motile. During ejaculation, sperm are propelled through the muscular vas deferens, which you can see in this diagram here. The vas deferens begin right after the epididymis. And then from there, the vas deferens combines with the duct of the seminal vesicle to form the ejaculatory duct. So there's 2 of these, as you can see here in this picture. And then the ejaculatory duct then exits into the urethra, which is then a single tube which exits through the penis. Semen is composed of sperm plus secretions of 3 sets of accessory glands. There's 2 seminal vesicles, which contribute about 60% of the total volume of semen. The prostrate gland secretes its products directly into the urethra through several small ducts. And the bulbourethral gland secretes a clear mucus before ejaculation that neutralizes the acidic urine remaining in the urethra. There's evidence that the bulbourethral fluid contains some sperm that's released before ejaculation, which may contribute to the high failure rate of the withdrawal method of birth control. The human penis is composed of 3 cylinders of spongy erectile tissue. During sexual arousal, the erectile tissue fills with blood from the arteries, causing an erection. The head of the penis, or glans, has a thinner outer layer compared to the skin covering the shaft and is more sensitive to stimulation. The glans is surrounded by a fold of skin called the prepuce, or foreskin. Now we'll switch to the female reproductive anatomy, starting with the external anatomy. The female external reproductive structures include the clitoris and 2 sets of labia. The internal organs are a pair of gonads, the ovaries, and a system of ducts and chambers that carry gametes and house the embryo and fetus. The female gonads are the ovaries. These flank the uterus and the abdominal cavity. Each ovary contains many follicles, as you can see here in the diagram. Each follicle consists of a partially developed egg called an oocyte, which is surrounded by support cells. An egg cell travels from the ovary to the uterus via the oviduct or fallopian tube. You can see the oviduct here in the diagram. Cilia in the oviduct conduct the egg to the uterus, which is also called the womb. The uterus lining is called the endometrium, which you can see in the diagram here, and this has many blood vessels. The uterine wall is very thick and powerful, consisting of lots of smooth muscle. The uterus narrows at the cervix, which opens into the vagina. The vagina is a muscular but elastic chamber that is the site of deposition of sperm during copulation and serves as the birth canal. The vagina opens to the outside at the vulva, which is the collective term for the external female genitalia, which consist of the labia majora, labia minora, and the clitoris. The labia majora are a pair of thick fatty ridges that protect the rest of the vulva. The labia minora are a pair of slender skin folds that surround the vaginal opening. The clitoris has a head called the glans, which is covered by a prepuce, which is a small hood of skin. The vagina, labia minora, and clitoris are become engorged with blood during sexual stimulation. The clitoris has many nerve endings, and therefore it's one of the most sensitive points of sexual stimulation. Sexual stimulation stimulates the vestibular glands near the vagina to secrete lubricating mucus. The mammary gland is an exocrine gland in humans and other mammals, meaning that it produces things that leave the body. They produce milk to feed young offspring. Within the glands, there are small sacs of epithelial tissue, which you can see here in the diagram, that secrete milk, which drain into a series of ducts that open at the nipple. The breasts also contain connective tissue and fatty adipose tissue. Gametogenesis is the production of gametes. Spermatogenesis is the formation of sperm, which is continuous and prolific in humans. Hundreds of millions of sperm are produced each day. Each sperm takes about 7 weeks to develop. Sperm are made in the seminiferous tubules, which are these long convoluted tubules within the testis. Now, if we look at a cross-section of one of these seminiferous tubules, we'll see something that looks like this. The sperm cells start off as immature cells near the periphery. And as they move down towards the lumen, they become mature until at the very end of the lumen, you have motile sperm that can move. And then these will continue their journey. They'll eventually end up in the epididymis, where they'll spend about 3 weeks. And during that time, they'll gain a longer flagellum and more mitochondria. The whole process actually starts in the embryo. Primordial germ cells in the embryonic testes give rise to spermatogonial stem cells through mitosis. Those aren't shown here in this diagram. But you basically would, at the very periphery here, you have spermatogonal stem cells, which divide mitotically to form a spermatogonium. So here's a spermatogonium. And each time they divide, one cell remains spermatogonial stem cells. So in other words, you'll never run out of these stem cells. The spermatogonium divides mitotically to form 2 primary spermatocytes. So you can see that here. They're just showing one, just to keep the diagram simpler. So here's a primary spermatocyte. So this division was mitonic, and therefore the spermatogonium and the primary spermatocyte are both diploid, meaning they have the full complement of chromosomes, which would be 46 in a human. Now, the next stage, each primary spermatocyte divides to form 2 secondary spermatocytes, and this division is by meiosis. So now we have the secondary spermatocytes, which are haploid. So in a human, they would have 23 chromosomes. At this point, though, these chromosomes consist of a pair of sister chromatids. So we still have sort of double the amount of DNA that we need to get to. So in the next step, each secondary spermatocyte divides to form a spermatid. And so now each spermatid has chromosomes that just consist of a single chromatid. And so we now have the correct amount of DNA. So it's haploid, and there's just a single chromosome. So 23 individual chromosomes. There's no pairing or anything like that. And then these spermatids are released. As you can see here, they're developing. And then they're released and as mature sperm cells into the lumen of the seminiferous tubule here. They travel through this seminiferous tubule into this other area and then into the epididymis, and they spend 3 weeks maturing into the final form that sperm take, which have a lot of mitochondria and a longer flagellum compared to the sperm that are initially generated within the seminiferous tubule. This is a diagram from your book showing a mature sperm cell. The head contains the haploid nucleus, shown here. At the tip of the head is a vesicle called the acrosome, which contains enzymes that help the sperm to penetrate the egg. Behind the head are many mitochondria that provide ATP for movement of the flagellar tail. Oogenesis is the development of a mature egg, and this is quite a prolonged process compared to the generation of sperm cells. Immature eggs form in the female embryo, but do not complete their development until years or decades later. The whole process starts in the embryo with primordial germ cells. These divide mitotically to first form the oogonium. Notice that this is a diploid cell. There are further mitotic divisions that lead to then the formation of the primary oocyte, which is present at birth. These cells begin meiosis, but they stop at prophase of meiosis 1. And these cells can be found within follicles, which essentially consist of this primary oocyte surrounded by support cells that protect them. Beginning at puberty, follicle-stimulating hormone, FSH, periodically stimulates some of the follicles to resume growth and development. Recall that the primary oocyte began meiosis 1 in the embryo. It now may complete meiosis 1 and become a secondary oocyte. So note that this is now has the haploid number of chromosomes. So this would correspond to meiosis 1. So the next step is the secondary oocytes begin meiotic division, but stop at metaphase of meiosis 2. And that's shown here. So now it started meiosis 2, but it stopped at metaphase. At this point, the oocyte is mature, and you have ovulation, you have rupture of the follicle, and the ovulated secondary oocyte. Now, if this oocyte encounters a sperm, the sperm can enter, and that actually will trigger meiosis 2 to resume. And then finally, you have true fertilization, which is strictly defined as the fusion of the haploid nucleus of the sperm cell with the secondary oocyte that has completed meiosis 2. One thing to note about these meiotic divisions is in producing the egg is that they're quite different than the meiotic divisions that led to production of sperm. In the producing an egg, the meiotic divisions are unequal in terms of cytokinesis, with smaller cells becoming polar bodies, and these eventually degenerate. So, for example, here with the primary oocyte, which is an arrested prophase of meiosis 1, it completes meiosis 1, but the cytoplasm is unequally distributed. So there's one large cell and one small polar body. Now this polar body will degenerate. Nothing will develop from it. And then in the next step, when you have the secondary oocyte, which starts to undergo meiosis itself, it ends at metaphase of meiosis 2. And then when it's stimulated by sperm entry, it then completes meiosis 2, and then you have a second polar body formed, and then you have the fertilized egg. And again, this second polar body would just degenerate, nothing would come of it. So this is quite different than the generation of sperm, in which you'd have a doubling of cells. So you'd go from 1 to 2, and then you'd have yet another doubling. So you'd end up going from 1 cell to 4 cells. Here you just go from 1 primary oocyte to 1 fertilized egg. Recall that in order to generate the secondary oocyte during ovulation, the follicle has to rupture in order for the secondary oocyte to leave. Once this happens, the ruptured follicle actually has a second purpose. It develops into the corpus luteum. The corpus luteum secretes 2 hormones, estradiol and progesterone. Both these hormones help to maintain the uterine lining during pregnancy. Estradiol helps to grow and maintain the endometrium thickness. Progesterone prepares the endometrium for the potential pregnancy after ovulation. It triggers the lining to thicken also, and it also helps trigger the acceptance of the fertilized egg. Progesterone also prohibits muscle contractions in the uterus that would cause the body to reject an egg. We're going to end this lecture looking at some differences between spermatogenesis and oogenesis. With spermatogenesis, during meiosis, there were equal divisions during meiosis 1 and meiosis 2 of the cytoplasm. And therefore, starting from 1 cell, you end up with 4 cells, which are all become sperm. Whereas with oogenesis, starting with 1 cell, as you go through the 2 parts of meiosis 1 and 2, you end up with just a single egg cell. And the reason for this was because of the unequal cytokinesis. So the cytoplasm was unequally divided, and you would have the formation of a polar body, which would degenerate and not go on to produce any more cells. The next difference is that spermatogenesis occurs throughout adolescence and adulthood. For oogenesis, this process stops around age 50. The next difference is that sperm are produced continuously without prolonged interruptions, whereas there are long interruptions in oogenesis. In other words, oogenesis begins very early in the fetus, and then it's interrupted for a long period of time, and then starts up again in puberty. And so there's this long gap, whereas with sperm, they're just sort of continuously generated.